Researcher is conducting a retrospective study. Okay, the study is going back in time, so it is known as a retrospective study. Fine. On breast cancer recurrence. Okay, records of number of patients on hormone receptor positive early stage breast cancer received adjuvant therapy with tamoxifen, drug of choice in breast cancer. Okay, all of you know it is an SERM, selective estrogen receptor modulator. Okay, clinical analyze okay, some of the relapsed patients had lower serum concentrations of endoxifen and 4-hydroxytamoxifen. These are the active metabolites. Okay, what can be the cause of drugs ineffectiveness in this subset of patients? First, you need to know about certain group of enzymes known as the Cytochrome P450 enzymes, which are a group of heme containing proteins responsible for majority of drug metabolism and are present predominantly inside the liver. They deactivate drugs, facilitate excretion, improve water solubility. However, they metabolize certain compounds to active forms also they can. Now what happens in these polymorphisms occur? Polymorphisms can occur in these genes coding, altering their expression or activity. Three important phenotypes exist. You know genotype, genetic constitution of an organism. Phenotype is the physical appearance of an organism. So poor, intermediate and rapid metabolizer. Identifying these three variations provides the framework for optimizing therapy. So tamoxifen, I have told you, tamoxifen is actually a prodrug. The prodrug which is metabolized, tamoxifen is actually a prodrug which is metabolized by CYP2D to its active metabolite, that is endoxifen. Patients with genetic polymorphisms result in poor CYP2D activity are exposed to decreased levels of metabolite. Then it will not be effective. Now this is required to treat hormone estrogen receptor positive breast cancer. And if there is a polymorphism, then decreased level of the active metabolite. The activation of downstream signal transducer proteins such as CRAS leads to activation of transcription factors that promote cell growth. Mutations or any change is associated with the development of colorectal and lung cancer. So this is obviously wrong. Then decreased activity of hepatic N-acetyl transferase results in diminished ability to metabolize drugs such as, yes, isoniazid and sulfonamides. The case of option B. So this is also wrong. Then thiopurine methyl transferase is responsible for the metabolism of thiopurine compounds such as the compound 6-mercaptopurine. This is metabolized. Okay. So if this is deficient, then there will be toxicity of 6-mercaptopurine, but this is not the case here. Then overexpression of P-glycoprotein in the tumor cells. I have already told you about P-glycoprotein. It is an efflux protein, right? Throws it out. So overexpression will be a cause of multi-drug resistance, overexpression. So we are left with the diagnosis of exclusion, polymorphism of cytochrome P450.